Welcome to The Explainer. Today we're looking at something that touches millions of lives all around the globe, and yet the whole thing is rooted in this incredibly tiny, microscopic cellular event. We're talking about trisomy 21, which you probably know better as Down syndrome. But instead of just viewing this strictly as a disease that needs to be cured, I kind of want us to look at this today as a really fascinating genetic mystery. We're going to unpack exactly how one tiny little variation in our genetic code can literally alter the entire blueprint of human development.
Okay, let's dive right into this. 1 in 700. That is the worldwide prevalence of live births for trisomy 21. That makes it, by far, the most common survivable chromosomal disorder on the planet. It's incredibly prevalent, and yet, the underlying biology driving the whole thing, it's astonishingly complex. So what exactly is it? Well, basically, trisomy 21 is a genetic variation caused by a triplication of chromosome 21. So instead of your typical pair, you've got a third copy hanging out in there. Honestly, the best way to think about this is like a volume knob on a stereo that's just been turned up a little too high. This extra genetic material alters the cellular gene dosage, and what that actually means is the body is overproducing certain proteins, which then sets off this whole cascade of changes affecting physical, cognitive, and overall systemic health. So here's our roadmap for today. First, what is trisomy 21? Second, we'll look at the genetic mechanisms. Third, the symptoms and clinical presentation. Fourth, prenatal screening and diagnosis. And finally, number 5, the really exciting stuff, the future of therapeutics. Understanding the cellular biology up front is really going to set up those astonishing futuristic therapies we'll talk about at the end.
All right, let's start with part 1. What is trisomy 21? Just a really brief medical timeline to get us situated.
To really figure out where we're going, we kind of need to zoom out first. If you look at the medical history of trisomy 21, there is this massive, almost century-long gap in our understanding. Back in 1866, British physician John Langdon Down became the first person to comprehensively describe the physical traits of the syndrome, which obviously eventually got his name. But since the tech literally didn't exist to peer inside the nucleus of a cell back then, the actual biological cause? Total mystery. That is, until 1959, when French geneticist Jérôme Lejeune finally spotted that extra chromosome hiding inside the cells.
Okay, moving on to part 2, the genetic mechanisms. We are zooming all the way down into the microscopic world.
Now, what's really interesting about this is, we usually talk about Down syndrome like it's just one single uniform thing, right? But actually, there are 3 completely distinct biological pathways that cause this overexpression of roughly 160 protein-coding genes. The vast majority of cases are caused by something called nondisjunction. Then you've got translocation making up about 3 to 4%, which is when an extra piece of chromosome 21 physically breaks off and hitches a ride on another chromosome. And finally, mosaicism at just 1 to 2%, which is when an error happens after fertilization, leaving the body with a mix of typical and atypical cells.
Since it makes up 95% of cases, let's look a bit closer at nondisjunction. This is essentially just a random cellular failure. During meiosis, the process that makes an egg or sperm, chromosome pair 21 is supposed to divide perfectly evenly. But here, they just fail to separate. So this leaves a gamete, which is usually the egg, with 2 copies instead of 1. And then, boom, when fertilization happens, the embryo gets a third copy from the other parent. And while this is a totally accidental error, the probability of it happening does correlate heavily with advanced maternal age.
Which brings us to part 3, symptoms and clinical presentation, the lived effect.
So what actually happens when every single cell in a person's body is carrying this extra genetic payload? The extra material subtly shifts development across the entire body. On the physical side, individuals often have upslanting almond-shaped eyes, a somewhat flattened facial profile, a single transverse crease across the palm of the hand, and generalized hypotonia, which basically just means low muscle tone. Cognitively, neurodevelopment is altered too, usually resulting in mild to moderate intellectual disability. But I really want to highlight something absolutely incredible here. Despite those cognitive delays, their adaptive functioning and their social-emotional intelligence are frequently highly preserved. That said, the systemic medical impacts can be really tough. Just think about this stark number, 50%. Half of all infants born with trisomy 21 have congenital heart defects. And the most common one is an atrioventricular septal defect, which often requires some pretty significant and immediate medical intervention. So, the crucial point here is how this genetic dosage impacts folks later on in life. It turns out chromosome 21 is the exact home of the APP gene, and that's the gene responsible for making amyloid precursor protein. Because folks with trisomy 21 have 3 copies of this gene, they chronically overproduce amyloid plaques, and tragically, this creates a direct link to early-onset Alzheimer's. In fact, over 90% of individuals will develop Alzheimer's-like pathology by the time they reach their 50s or 60s.
All right, let's jump to part 4, prenatal screening and diagnosis, the clinical reality.
Because these systemic effects are so profound, catching these genetic markers early on is a big deal. If we guide our focus back to the very beginning of life, modern obstetrics has some pretty amazing tools that cleanly distinguish between risk assessment and definitive medical confirmation. Screening tests basically give expecting parents a probability, with 0 risk to the pregnancy, just maternal blood and ultrasounds. On the flip side, diagnostic tests give you definitive chromosome confirmation, but because they involve invasive tissue or fluid extraction, they do carry a slight risk of miscarriage.
Here's how that tiered approach actually looks in practice. First, you have your standard first trimester screen for risk assessment. But the absolute game changer here is NIPT, non-invasive prenatal testing. This is a massive breakthrough because it isolates cell-free fetal DNA directly inside the mother's bloodstream as early as week 10, and it boasts a greater than 99% detection rate. If a screening does come back high risk, parents can then opt for those invasive diagnostic tests, like CVS, where they extract placental tissue, or amniocentesis, the absolute gold standard, which uses amniotic fluid to look right at the chromosomes themselves.
And that brings us to our final section, Part 5, the future of therapeutics, the horizon.
For the longest time, getting a trisomy 21 diagnosis meant preparing for an entirely unchangeable reality, right? Because that extra chromosome is literally in every single cell. But wow, peer-reviewed breakthroughs from just 2025 and 2026 are completely redrawing those boundaries. The rapid pace of these discoveries is just staggering. We are talking about genetic engineering and targeted therapies going after the root causes at unprecedented speeds. We're not just talking about managing symptoms anymore. Scientists are figuring out how to intervene at the molecular level.
For example, take this astonishing milestone study by Hashizume and colleagues in PNAS Nexus. They used the CRISPR-Cas9 system to target and literally chop up only the 3rd extra copy of chromosome 21 in living human cells, and they managed to leave the other 2 healthy chromosomes completely untouched. This concept of trisomic rescue in laboratory stem cells proves that, technically speaking, physically removing a pathogenic chromosome without harming the healthy pair is entirely viable. Normal metabolic function was completely restored. It's just wild.
Let's move to the next breakthrough and see how this builds on that. This is a totally different but equally brilliant approach. Instead of trying to delete the chromosome, scientists essentially hijacked a natural biological process. See, females naturally turn off one of their 2 X chromosomes using something called the exist gene. So, researchers used CRISPR to insert that exact gene right into the extra copy of chromosome 21. And guess what? The gene produced an RNA that coded that 3rd chromosome, turning it into silent heterochromatin. It beautifully corrected the dosage imbalance without having to delete a single thing.
But wait, what about folks who are already living with Down syndrome? Enter small molecule therapies. The 2026 ICOP project is running cognitive trials on a drug called AEF0217, which actually normalizes overactive cannabinoid signaling in the brain to improve memory and functional independence. At the exact same time, researchers discovered that restoring a depleted molecule called pleiotrophin in adult models physically rewired faulty neural circuits. These breakthroughs are monumental because they completely shatter the long-held dogma that cognitive changes have to be treated before birth. This offers immense hope for adults right now.
We've traveled all the way from the historical discovery of an extra chromosome to literally silencing it at the molecular level, which leaves us with a truly profound question to ponder. If we can eventually silence or delete an entire extra chromosome safely in human beings, how are these breakthroughs going to redefine the very concept of chromosomal disorders in the decades to come? Thank you so much for joining me for this explainer. I really hope this deep dive leaves you just as fascinated and eager to track this incredible medical frontier as I am. See you next time.